Alright, so this is DNA transcription. It's pretty much the same for prokaryotic cells and eukaryotic cells. Right here, this is pretty much the big picture stuff. So, like the initiation, elongation, and termination is pretty much exactly the same for both of them. So, initiation is pretty much the first thing that happens, and that's when an RNA polymerase pretty much binds to a promoter. And a promoter is the sequence of DNA right there, and this green thing is the polymerase. And the part of DNA that's about to be transcribed is the transcription unit right here. And initiation is pretty much that bonding together, and the DNA helix, this is actually a helix right here, and that's unwinding as it goes through, and nucleotides are being added. So this is where they start getting added right here with the RNA transcript. And the template strand is the DNA that is about to be transcribed into mRNA. And the one thing that's different about initiation in prokaryotic cells and eukaryotic cells is that in eukaryotic cells, the the TATA box is where that's pretty much like a starting kind of thing, and that's where the polymerase bonds to the DNA. Alright, so this is elongation, this part right here. And so nucleotides are being added to this 3' end, and it's going that way. So that means it's growing from the 5' end to the 3' end, but it's just kind of being pushed out, and it's about to be ejected eventually like that. So in mRNA, the A and T bond together, and everything bonds together just like DNA. But the only difference is that for instead of thymine, it's uracil right here in the mRNA. So what initiates this RNA transcription is AUG. And what stops it is the UAA right there. Let's see, what else is there? Okay, well, moving on to termination. So termination is when the mRNA comes out, the RNA polymerase falls off, and another difference between prokaryotic cells and eukaryotic cells is that this mRNA is usable in prokaryotic ones, and it's not usable until after it's kind of been messed with a little bit for eukaryotic cells, which I'm about to get into in a second. Alright, so this is the pre-mRNA processing, which is only in eukaryotic cells. So what happens is a 5' cap will be added, which is it's kind of just like a G that's kind of been messed with a little bit. And also a poly A tail is added onto the end of it. And what these help with, they are right here. They facilitate export mRNA from the nucleus. They protect from hydrolytic enzymes, and they help ribosomes attach to the 5' end once it gets into the cytoplasm. So the poly A tail is just like a bunch of A's together, like 50 to 250. And then there's these other things called UTRs, they're untranslating regions. And like not all of the mRNA is translated, only a little bit of it is, actually. But they help with other things in the cell. And so some of the stuff that doesn't get translated undergoes this thing called RNA splicing, which is like a removal of like a ton of mRNA that doesn't come into like amino acids. So the parts that do get translated are exons, like on this end and right there, and then the intron isn't shown externally. So SNRPs and other proteins come together to make a spliceosome, which helps cut out the intron. And so that's like a cutout intron. That's the exons without the intron, and the spliceosome components come off. And that's pretty much it.